Thank you to the organizers for inviting me to present on the diagnosis of active TB, which I've given the subtitle Modern Tools for a Post-Pandemic Response. My name is Morten Ruhwald. I'm the Director of TB at FIND, the Global Alliance for Diagnostics. So this is the first of 3 TB presentations at this conference. So I'll give a little bit of context on TB. So if we ignore COVID-19 for a second, TB remains the largest infectious disease killer. The disease develops slowly, it's difficult to diagnose, and it's hard to treat. Most cases are found among the urban poor, and these are probably where we have some of the largest diagnostic gaps. And COVID-19 really has impacted TB services and also the health-seeking behavior across the world. But there is a global push for post-pandemic recovery, but we need new tools and new processes to make this happen. And I'll try to cover this as part of my presentation today. Just the numbers, we have 10 million people who get TB every year. 1 million of these are people living with HIV. 500,000 have drug-resistant TB. We have 1.4 million deaths. We have 4.2 million who remain undiagnosed every year. So that's almost half of all TB patients. Globally, only about 1 in 3 TB patients are bacteriologically confirmed. Only 20% are diagnosed with a WHO-recommended molecular diagnostic test. And only 1 in 3 of those who have drug-resistant TB are tested and put on relevant treatment. So as you can see, there's huge diagnostic gaps. COVID only made things worse, and the first 12 months of COVID-19 seems to have eliminated 12 years of progress in the fight against TB. We've lost 1.3 million people who are not notified in 2020 compared to 2019. And we have seen the biggest impacts in the high TB incidence countries, in India, Indonesia, Philippines, China, Bangladesh, but it's a global phenomenon. As you can see here, the impact on mortality is anticipated to be long-lasting. The incidence is increasing, and it will also be a long-lasting effect. Diagnosis remains the weakest link in the cascade of care, and we really urgently need to have better tools. The reason why this is not perfect is that the tools we have are not fit for purpose. People initiate care where there's no capacity to diagnose TB, so we need tools that can go to that level of the healthcare. And we do a lot of symptom-based screening, which really probably misses half of TB cases in the community. And there's a big reliance on sputum, which also makes the diagnosis difficult. And we do perhaps select patients who have the most advanced disease. But COVID also has come with a lot of new opportunities. We've seen a game changer in the way, diagnoses have been brought closer to the patients. Many people are very comfortable now with self-testing. We've seen a massive investment in the diagnostic portfolio. A lot of new molecular platforms are coming out. And it's really up to the TB community now to leverage the investments, both in the infrastructure, the digital, the connectivity, and also the new tools, to see if we can build back better in a new and perhaps a new reality with new tools. And this is not only for active TB, there's also a lot of stuff happening for TB infection and test and treat strategies. So I'll talk about that over the next 25 minutes. The first class of technologies I'll talk about is chest X-rays and computer-aided diagnostic software. So chest X-rays remains the most sensitive tool we have to detect long TB. We have digital X-rays that now can be fed into artificial intelligence software, and within a minute or so come with a diagnosis or an abnormality score at least. We have at least 15 products that are either commercially available or close to becoming commercially available. A lot of version upgrades, a lot of new features being added all the time, and it's really a new area for policymakers and regulators. Together with Stop TB, FIND have tried to bring a little bit of order in this field through the AI for Health webpage, where you can then look at the different softwares head to head and make the procurement decisions there. Not all softwares are created equal. And this is a very nice study from Vietnam that has looked at more than 1,000 participants, whereof more than 100 had confirmed TB. You can see in these rock curves here that some seems to be very good, where others are not working at all. And some of those that are performing better are even performing better than expert radiologists with more than 30 years of experience. And then we have the intermediate training readers here with about 5 years experience, and many of these softwares are outperforming them as well. So clearly we need much more evidence like this to drive sort of the comparisons. This becomes very powerful when you combine it with portable X-ray systems. And the smallest ones, like this one from Aspen Imaging in the US, you have a super sensitive screen and a very small and portable X-ray generator. You then put it on a tripod like this, and then you take your images. They are low power, so you don't need to have a lead apron as long as you are a couple of meters away from the instrument. This is another instrument where they've implemented it in a really rural mining community in Pakistan, and you can see this battery-operated tool really does deliver. This guy is sitting with the software here next to the X-ray, and they have sort of a unit of a couple of people that can screen perhaps up to 200-300 people a day. These are with solar powered, so you can really take it into the communities where there's no electricity or in the really rural settings. You can carry them in backpacks or put them in trucks, etc. FIND have written or generated a report where you can read about the various different types of software, but also on the hardware, and you can find it at the FIND webpage. Showing you here a small movie about how a screening is done. He's being questioned, and they fill out the questionnaire. Then the chest X-ray is done simply like that, and then the image goes in here and gets interpreted on the other X-ray. And with a 5-minute workflow, you have screened an individual that can either go for further confirmatory testing or being that go back into the society. So there are several other technologies: point-of-care ultrasound, digital-enabled stethoscopes, where you or AI-enabled stethoscopes, or even AI-enabled cough apps, where you cough into your smartphone. All of these approaches are being developed, and you could see, if you had a screening tool that basically is just a recording of a cough, that would be a complete different way of looking at TB. The next class of technologies I'll talk about is LAM tests that really bring diagnostics closer to where the patients are. And this is of high relevance for HIV. So LAM is a molecule that's found in the outer membrane of the Mycobacterium tuberculosis. It's found in the granuloma, or where the site of infection deep in the lung. And it then gets secreted into the bloodstream and gets secreted into urine, where it can be picked up. So we have the ALEA-LAM and we have the Fuji-LAM as the established tests at the moment. And the Fuji-LAM seems to be at least or about twice the sensitivity at a comparable specificity as the ALEA-LAM. And the unique thing about this class of diagnostic is that the poorer the immune status of the patient, the better this test seems to work. So in participants with a really low CD4 cell count, those with advanced HIV disease, the sensitivity is almost 90% here with the Fuji-LAM, and it is retained to about 45%, 43% here around with the high CD4 cell count. The ALEA-LAM has about 56% sensitivity in that population, and it drops down to about 11% with a high CD4 cell count. So there's a lot of anticipation around the Fuji-Lam test being able to deliver where, you know, a better diagnosis for this underserved patient population group. In the HIV uninfected, and that's where we hope this class of technologies can go eventually, is we're seeing a sensitivity of around 50%, where the Lam has only about 10% sensitivity. So clearly, you know, this is not good enough, but we have good proof of concept that we can make this better. And we're working on 3 different approaches: better reagents and pre-analytics, better sample preparation, also pre-analytic, and then for the analysis itself, better assay design. So we're working together with many other partners in developing new reagents, new antibodies, etc. We're working on ways to concentrate the Lam molecule in the sample. And we are also working on new innovative assay designs with many different manufacturers using readers or various ways of amplifying the signal. This is just one example of how the future might look for Lam-based assays. This is the project from SD Biosensor that recently was kicked off. So they aim to develop a reader-based tool here with a fluorescent dye. And then they have a suite of different readers that you can then put the test into. Some of these are battery operated, and they all offer connectivity, and we anticipate these to be ready for trialing sometime in 2023. I've been asked to talk mostly about active TB diagnostics and molecular tools, and this is where we are at the moment. We have for sputum-based tools, we have microscopy, we have line probe assays for drug resistance detection, we have LAMP assays for really simple TB detection, culture systems, and then for the molecular, we have the mobile TrueNAT system or the GeneXpert system. But it's clear that, as I mentioned in the introduction, only 1 in 3 patients are bacteriologically confirmed, and only 1 in 5 patients are diagnosed with molecular tests. So what we're offering at the moment is not good enough. And there was a recent systematic review commissioned by WHO that basically concluded it was uncertain whether there had been a reduction in mortality with the implementation of the expert MTB RIF compared to sputum smear microscopy. And the reasons are many and demonstrated in many, many different studies, and it's basically a poor fit for the healthcare system. So we need something that's simpler. So we need new sample types. We need new, simpler assays like point-of-care molecular assays, or we need really better sample transport systems for centralized high-throughput molecular tools. And I'll start talking first about new sample types. There's been a lot of work on swab-based diagnostics through COVID, and these are also, when you scrape the back of the tongue, we are starting to see good evidence suggesting that this could be a viable approach for non-sputum-based diagnostic for TB. This is just one of the new swab types that have been developed with funding from the Gates Foundation together with Quantigen and Stereopack. They've developed a dry swab here. You self-swab or with help from a healthcare provider, and then you put the dry swab into this media-free transport, this cartridge, you could say. You sort of plug it in there, and then you can send it to the lab, where they can automatically decap here and add different buffers, etc. And then you can either use a centralized or point of care testing with this kind of tool. I'll talk about it in a minute again. We've also seen a lot of work on aerosol-based capture, either where you blow into a tube where there's a small filter that kind of captures the bacteria, or in face masks where you have absorptive strips where the bacteria or the viruses kind of gets captured on these strips, And you can then take the strips out and use a molecular assay like the GeneXpert system to detect TB there. This project is even more futuristic. So this is a wearable, instrument-free, CRISPR-based tool that has been developed for COVID-19. So you wear the mask, and inside the mask there's this sample collection zone here, where the viruses are then captured. And you then press this blister on the side here. It washes through this sample collection zone, gets into a lysis zone where the viruses, in this case, are being made available for the genetic material in the viruses is being made available for analysis. And then they have an isothermal amplification and the Sherlock detection zone, and it goes out on a lateral flow here that is in the side of this device. Even more futuristic, you see this this chip-based approach where the assay is also captured here, but these are prototypes in development. I'd also like to mention that we now have WHO policy on stool as a sample for pediatrics, so another non-sputum-based sampling method that really is needs to be now taken up in country after demonstrating good evidence for its use. And you can look here at the one of the methods, the SOS method developed by the KNCB group in the Netherlands. They have a full standard operating procedure and demo videos to support how to use these. Then we have new molecular tests. Currently, WHO approved our Cepheid system and the MortBio. Recently saw the 10-color instrument and the XDR cartridge from Cepheid, but we also have from MortBio the true new RNA fluoroquinolone and bedaquiline chips, and also TB and COVID chip in development. There are two new large platforms designed basically to go into the space where Cepheid is dominant at the moment. The standard M10 from SD Biosensor and the RNQ from Bioneer. Standard M10 comes with up to 8 random access modules, a nice assay panel. They can use either isothermal or qPCR. It can take up to 12 targets. And the TB assay, which is CE marked here in Q4 2021, has RIF and INH inside the cartridge and gives a result within 60 minutes. The Bioneer has up to 48 targets, so it offers basically TB, RIF, INH, fluoroquinolone, and second-line injectable detection in the cartridge, and I hope they will be working on perhaps a more modern panel where perhaps the second-line injectables is substituted with a more relevant drug. This instrument here offers results in less than an hour, and really seems to be a very powerful tool with a nice assay menu as well. But where we probably need to go is point-of-care molecular, and many of you are probably aware of the EXPEL trial that was published recently in New England. And here they demonstrate that if you place the diagnostic next to the patient, you can get a 56% increase in treatment starts measured at day 40 compared to standard of care. They demonstrated this with the Cepheid Edge system. We had anticipated the Cepheid Omni to be launched, but this has been cancelled from Cepheid's side. So there's not going to be an Omni platform for TB. But we also have MortBio that could go into this use case as well. But there is a lot of fast followers, and I'll talk more about the Lumira technology in a second, but SD Biosensor could probably also be pushed really lower into the healthcare system. But through the investments from COVID, we're seeing a lot of really simpler platforms coming through. And I'll talk also about those in a second. So the first one here is the Lumira DX. It's a platform that is anticipated to be put into clinical trial in about a year's time. They go straight for a swab as the sample. You have a simple lysis step, it takes about 5 minutes, and then you put it on the strip here and put it inside this smart little machine that can both do immunoassay and molecular detection. And within 15 minutes, they claim to have the amplification done, and a total time to result is about 20 minutes and a non-sputum-based tool. But COVID has led to a lot of other platforms that are coming through. I'm just giving you 2 examples here. So this is the Lucira COVID test. It was launched about a year ago. It's a single-use, over-the-counter molecular COVID-19 self-test. So you basically, you do your COVID swab, put it in this instrument, it does the PCR, and then you throw it away afterwards. We are also seeing CRISPR-based technologies really being accelerated through the COVID pandemic. So this is from Tata in India. So it's made in India device that really is super simple for COVID detection. And we would anticipate stuff like this coming in for TB as well. But there's a lot of other platforms that are happening, and some of these already have TB projects. So I think we should anticipate much more diversified environment for diagnostics in the future, which is fantastic. So now I'll talk about the centralized high throughput approaches. So we have now 4 WHO recommended centralized assays and a couple of others that are not recommended yet. But all of these platforms offer high throughput testing, upfront NIH, isoniazid testing, and they can be used for multiple diseases like HIV, hepatitis, etc. The performance is similar to expert, and the resistance detection is for isoniazid RIF is similar to line probe assays, and they have all been policy recommended. So you can imagine a future where you have community healthcare workers that do swabbing of a large number of patients. You have a proper sample referral system, and then you have these centralized approaches. So you can run up to 384 specimens in an 8-hour shift. And if you then have a proper connectivity tool that allows the diagnosis to go back to the patient or the healthcare workers in the community, you really have something that's different from where you are now, where the patient has to go into the level 2 to get the sample done. But with new tools like this one I talked about earlier, this self-swapping device developed for COVID, and new tools, you can really go into a much more industrial scale here. So this is a workflow that's been developed by the Gates Foundation with Quantigen, Stereopac, Corteva, and LGC under the for COVID. They now have a fully automated workflow, and I'll just walk you through how that looks. So they do an automated sampling, swab handling, and sample preparation. Then they run it on the Nexa high throughput PCR system, and it can do up to 100,000 samples per day. This instrument, so this is really, really high throughput. They use an assay tape with 6,000 samples per run, and then this tape, you can see the reel here being assembled, is then undergoing PCR or thermal cycling, and then you do a reading afterwards. Because there's a barcode on this fancy device here, you really ensure that the sample can be tracked back, or it's barcoded throughout the analysis and the handling. You can really make sure that few or no errors are done, and the result gets back to the relevant patient in a fully automated way. Also want to mention that 2022 is the year of targeted sequencing for TB. We have now fully sort of end-to-end workflows that will be going for WHO review. I'm naming some of the manufacturers here on the assay side and on the sequencing side, and giving an image here of one of the smallest sequences of the nanopore sequencer from Oxford Nanopore here, just to give you an example of how simple it can be, or how how small these instruments are becoming now. And I'll finish off by just mentioning that we have more than, switching gears, and now mentioning that we also have now 15 products entering the market for TB infection testing. Some of these are for high throughput. Some of these are very similar to the quantiferin test. Some of these are rapid diagnostic test kind of layouts where you, of course, still have to incubate the sample, but then you can, with a really fast rapid diagnostic test, diagnose TB infection. And then in 2022, we also anticipate several new specific skin tests that uses the same antigens as the IGRAs to deliver IGRA-like performance in a skin test format. So my take-home messages after this rundown of quite a lot of technologies has been, or is that we have a growing pipeline of connected new diagnostics and new sampling strategies. And I think we will come into a future where we will see a diversification and the end to the one-size-fits-all approach we've had in TB. We'll be moving away from sputum, we'll be moving towards point of care, and we will have a lot of digital diagnostics and connectivity-enabled diagnostics. However, there is, as I started by saying, there are major gaps that remain and needs to be tackled. We have tools already now that are not being used, and we can reimagine triage and screening approaches with these tools. And we urgently, of course, need new tools to protect the many new TB drugs that are coming out. So molecular and drug susceptibility testing should be key themes to pursue. I'll finish off just by thanking the donors and the fine team in Geneva, but also the fine team in our country offices who are supporting a lot of this work. Thank you very much.